I'm introducing myself. I'm Peri Tobias. I'm just going to give an overview on the work that we're doing collaboratively on working towards understanding the host pathogen interactions. So really, we are, what we've been doing the last few years at Sydney University is working across a large number of collaborative projects and trying to understand this molecular interaction between the plant and the pathogen. Basically, we're doing that through our collaborations, and that's with New Zealand, with Brazil, with South Africa, and anyone who will work with us. This Melaleuca quinquenervia leaf is on a plant just down the road from me. You can see that it's just has absolutely no resistance response. I think in the cereal rusts they talk about green islands. It's a parasite, this fungus, so it actually manipulates the host to keep it alive so that it can feed itself. And I think that's a very nice leaf to show that example of how the plant is really just providing food to the parasite. So we work on rust pathogen genomics and transcriptomics to try to understand the basis of the pathogenicity. We're hopeful to look for differences across strains and also to look at pathogen population structures and observe changes in virulence. So these are things you can learn from the genomics and transcriptomics. I wanted to highlight this. The Plant Breeding Institute, as Robert's been involved in for many, many years, when the incursion of myrtle rust came to Australia, one of the initial projects that he and Karanjit Sandhu were working on was actually collecting those early isolates and getting single pustules and increasing them, and then developing a bit of a collection of those. So we've got spore collections. So you saw that image of Robert's earlier, the liquid nitrogen. So we have our first incursions sort of databased in this storage, which is great. So we can go back into these rust collections and look for changes that might be happening. So I'm not sure, the one that we work a lot on is AU3, which is the one that we've actually done a lot of our downstream work on. And in fact, I collected one of those initial samples from Queenscliff, New South Wales, so I was there in the beginning as well. Robert initiated the long read assembly for the genome, which was really started in 2016 and then ended up being a massive collaboration across with Plant and Food New Zealand people and ANU. And we assembled the genome and did some really interesting analysis and found how large the genome for this little pathogen, this tiny spore, is as 1 gigabase genome, and largely shaped by transposable elements. These are these sort of repetitive regions of DNA which can move around and maybe increasing the virulence. In 2019, we had Carolina Hayashibara from Sao Paulo came and worked with me on some comparative work on effectors, and we tried to look for differences across some sequences of effectors. Effectors are the particular manipulating genes from the pathogen that manipulate the host. And so these are predicted effectors, and she developed some primers and screened a number of different strains of Ostropaccinia acidii, and we did see a little bit of difference, but then just more recently, Louise Shuey and Flavia Bonora have just been running the same set of tests, and we don't see a lot of difference in these particular regions that we initially chose based on the sequences. So that's not a good clue to identify strains, but yeah, that was something we were testing. More recently, published the preprint of the revised genome because we're now able to do a lot more comprehensive genome assemblies. And again, a big collaborative group of people who you will hear from some of them during the conference. So Tejas Bourfleur and Zhenyan Liu and Benjamin Schlesinger, Ashley Jones. They will be talking later. Peng Zhang from the Plant Breeding Institute worked to try and visualise the chromosomes. Once we'd worked out that we had 18 chromosomes in the genome, she was able to visualise them, and so that was pretty interesting, and we're still working on this project. Last year we were able to identify that the particular mating structure for the pathogen, and so I'm going to run out of time. I think I'm talking too long, but what was interesting here was to see that we could identify some difference in one part of the mating locus of the genome. So this looked like a promising target for getting some variation to identify strains. We also work on the host, so we're working on some host genomics and transcriptomics. So we're looking at the molecular basis to infection and also the molecular basis to effective response in resistant plants. And we're doing this also with the genomics using, I think this is kind of just an example of how the technology has advanced so fantastically in the sequencing and also in the computational software, so that we can actually pull apart the genomes from the two parental plants within the single nucleus. That's the DNA, that tangled mess. Bioinformatically, we can start to pull it apart and build chromosomes. This is all computationally, and then we can start to actually drill down and look at genes that might be conferring resistance. And so this year we've put a preprint up on the Melaleuca quinquenervia genome, and we did it in that manner. So we've pulled the genome apart, and that's going to be discussed further by Stephanie Chen and Elissa Martino, who were the first authors on this paper. And I'd just like to mention that Yan, who I mentioned earlier, had a very big role in developing this pipeline to annotate disease resistance genes, and so this is now under review. We've got a RenSec project happening still, which is looking for resistance genes across different plants and in different phenotypes. So these are the DNA is pooled, and we can start looking at whether there are any particular resistance genes that are more present in resistant versus susceptible phenotypes for these species that we're testing. So that project was really a bit halted during COVID, but now is starting to build up again. And that's with Sam Bassavim, Peri Annan, and Louise Shuey. Dear to my heart is this little plant, Syzygium lemanii, because I worked on it for my PhD. And I've been screening a group of these plants for the last 4 to 5 years now, and they consistently show either highly susceptible or highly resistant. And I just point out that this is a resistant plant, and every time I inoculate it, it gets these little hypersensitive response flecks indicating zones of dead tissue. So those cells are dying wherever the spore lands and tries to germinate and infect. This other plant is always susceptible, and these are actually field infected. So this is actually out in the field, even though when they're growing, when we control inoculate, they have the same response, but even in the field they do this. So bioplatforms have funded some sequencing, and we're doing some, again, pulling those genomes apart from scaffolding them into chromosomes. And right now we're running this plant through MinION. In fact, Jacob Downs has been running it just over the last few days, so we'll be able to do some comparative work on those genomes. I've been working with a very large group in biosecurity, looking at remote sentinel spore trapping, capturing spores through this Burkhardt sort of suction thing that takes the spores and puts them into little tubes. We send the tubes off to have the DNA extracted, So this is a very large group of people from different organisations, and we've been working on this project as a pilot to see if we can detect detect presence, absence, and detect. So we're using qPCR as well as metabar coding. And again, there will be further talks on this, so I won't go into any further detail right now. As well as that, because we have these spore isolates that we have been collecting, we can run phenotyping tests really well. The serial rust setup is fantastic at the Plant Breeding Institute, so we've got a really nice setup that was transitioned to work with myrtle rust. And we phenotype naive host plants, and that hopefully will inform management practices, and also potentially for diagnostic assay of where we should be putting resources. So we've done, since 2021, Alyssa Martino has been screening around 15 WA naive hosts, Melaleuca species, various species. We also did some Melaleuca alternifolia. I noticed it was Leptospermum alternifolia on a previous presentation, but that's the tea tree oil industry. Leptospermum scoparium we've screened, and Melaleuca quinquenervia and Syzygium lemanii. And Alyssa published a paper last year on the WA screening that she'd done, and I think there's a whole bunch more that she's done since then. Okay, I think that's it then. This is a single pustule, an image of the spores in a single pustule, and you can see how many spores are produced just from one pustule, and they're airborne. It's very, very hard to control this pathogen. So I'm happy to take questions, but I also advise that a lot of these this is an overview of what we're working on, and most of these will be presented in much greater detail throughout the program.